Our immune system is a powerful defense force, protecting us from harmful invaders like bacteria and viruses. However, sometimes this protective shield can turn against our own body, mistaking our cells and tissues for enemies. This misguided attack is called autoimmunity, and it can lead to a range of diseases. Type 2 hypersensitivity is one such example where our immune system goes awry. In this case, the culprits are antibodies, special proteins that normally target foreign invaders. In type 2 hypersensitivity, these antibodies mistakenly bind to our own cells and tissues, triggering a cascade of events that result in inflammation and damage. Imagine our immune system as a security system programmed to identify and eliminate intruders. In type 2 hypersensitivity, this system malfunctions, mistaking the house's furniture for burglars and launching an attack. This friendly fire can have serious consequences for the house, just like type 2 hypersensitivity can damage our bodies. This type of immune reaction is implicated in several diseases where the immune system attacks specific tissues or organs. Understanding how antibodies and the complement system contribute to type 2 hypersensitivity is crucial for developing effective treatments for these conditions. Antibodies, also known as immunoglobulins, are the foot soldiers of our immune system. Produced by B cells, these Y-shaped proteins patrol our bodies, searching for foreign invaders like bacteria and viruses. Each antibody has a unique binding site that recognizes and latches onto a specific target called an antigen, much like a key fits into a lock. In type 2 hypersensitivity, antibodies mistakenly recognize our own cells and tissues as foreign. These misguided antibodies bind to antigens on the surface of our cells, essentially tagging them for destruction. This binding triggers a series of events that lead to the activation of a powerful weapon in the immune system's arsenal, the complement system. The complement system is a group of proteins that act like a chain reaction, amplifying the immune response. Imagine a line of dominoes. When the first one falls, it triggers the next, and so on, creating a cascade effect. Similarly, when an antibody binds to an antigen, it activates the first protein in the complement cascade. This activation sets off a chain reaction, activating other complement proteins in a specific sequence. This cascade of protein activation leads to the formation of a membrane attack complex, or MAC, on the surface of the targeted cell. The MAC acts like a hole punch, creating pores in the cell membrane and causing the cell to swell and burst. Section 4 Inflammation Unleashed. C3a and C5a call in the troops. The complement cascade doesn't just directly destroy cells, it also triggers a powerful inflammatory response. Two complement proteins, C3a and C5a, act like distress signals, attracting inflammatory cells like neutrophils and macrophages to the site of antibody binding. These inflammatory cells are like first responders rushing to the scene of an accident. They release a barrage of toxic substances, including enzymes and reactive oxygen species, in an attempt to eliminate the perceived threat. Think of it like this. The antibodies raise the alarm, the complement system calls for backup, and the inflammatory cells arrive ready to fight, causing collateral damage in the process. Section 5. Fc receptors, another path to inflammation and damage. In addition to activating the complement system, antibodies can also trigger inflammation and tissue damage through another pathway, Fc receptors. These receptors, present on the surface of immune cells, like macrophages and natural killer cells, bind to the tail end of antibodies. When an antibody binds to an antigen on a cell surface, its Fc region sticks out, acting like a beacon for Fc receptors on immune cells. This binding triggers the immune cells to engulf and destroy the targeted cell, a process known as antibody-dependent cell-mediated cytotoxicity, or ADCC. Fc receptor binding also activates these immune cells, leading them to release inflammatory mediators that further contribute to tissue damage. Section 6. Good pasture syndrome, when antibodies attack. Good pasture syndrome is a prime example of a type 2 hypersensitivity reaction where antibodies mistakenly target a specific part of our body. In this case, the antibodies attack a protein called collagen 4, which is found in the basement membranes of our kidneys and lungs. These misguided antibodies bind to collagen 4 in the kidneys and lungs, activating the complement system and attracting inflammatory cells. This immune assault damages the delicate filtering units of the kidneys, called glomeruli, and the tiny air sacs in the lungs, called alveoli, leading to kidney failure and lung bleeding. Good pasture syndrome illustrates the devastating consequences of type 2 hypersensitivity reactions, highlighting the importance of understanding and controlling these misguided immune responses.